Chapter 24, Carboxylic Acids and Esters. And here we see a picture of orange juice. And oranges, and there's limes and lemons in the background, are citrus fruits. And citrus fruits are classified as citrus fruits because of citric acid, which we're going to see is a tricarboxylic acid. For the carboxylic acids, the molecules end in this moiety. You have a carbon that has a double bond to an oxygen and then an OH. And that is also, you can write it COOH or CO2H. These electrons are delocalizable. So we know from our Lewis dot structures there'd be two lone pairs. This pair can come here, pushing that pair there. And so this form is in resonance with this form. Because the negative charge in that oxygen, if the hydrogen pops off because it's acidic, let's say the H plus pops off like acids do, the negative charge can be stored on both oxygens. You have this form, then you have this form. And because that negative charge is being shared on two oxygens, that makes that way more stable than an O minus. And so an alkoxide, just a RO minus, is super basic, whereas the ion of a carboxylic acid is not very basic at all, making this quite an acidic compound. So this is why carboxylic acids are a lot more acidic than alcohols. The hydrogen doesn't really pop off of alcohols very easily because the resulting RO negative alkoxide is extremely basic. Whereas here, the resulting anion is not very basic, and therefore this makes this a much more acidic compound. So carboxylic acids are acidic. You always assign carbon 1 to the carboxylic acid carbon. And that is actually the highest priority functional group. So even if you have other functional groups in there, the carboxylic acid still gets to be carbon number 1. And then we drop the E from the alkane ending and you get oic acid. Ethane becomes ethanoic acid. Methane becomes methanoic acid, also called formic acid. Propane becomes propanoic acid. And ethanoic acid is also called acetic acid. That's a common name. And again, as you move away from the carbonyl carbon, the first one is called the alpha, then beta, then gamma, then delta, and then we have epsilon here. So here we have alpha hydroxy butyric acid, because you have butyric acid, 1, 2, 3, 4 carbons. And again, the carboxylic acid is carbon 1, and at the 2 carbon you have the hydroxy group. So this is either 2-hydroxybutanoic acid or alpha hydroxy butyric acid. Here we have 2-aminopropanoic acid or alpha-aminopropanoic acid. Again, the alpha carbon is the one right next to the carbonyl carbon. And that was true with aldehydes and ketones also. And here we have 3-chloropropanoic acid. Again, carboxylic acid is carbon 1. And this chlorine is attached to this carbon here. So it's either the beta carbon or the 3 carbon. Both names are correct. So write the formulas for 3-chloro-4-methyl-hexanoic acid. Well, we first write the hexanoic acid. We need 6 carbons. So here we have our hexanoic acid. Then at carbon 3 there's a chlorine group, chloro, and then at carbon 4 there's a methyl group. For alpha-aminobutyric acid, we first draw our butyric acid, a 4-carbon carboxylic acid, so butanoic acid, both names are usable. And then at the alpha, or the 2 carbon, the one right next to the carbonyl carbon, we have our amino group. Alpha-aminobutyric acid, or we could call it 2-aminobutanoic acid. Both would be correct. So, and again, here we have some common names because we've been seeing 2 names for different things. Methanoic acid is formic acid, and they're made by ants. Ethanoic acid is also called acetic acid. So a common name in chemistry is chloroacetic acid or trichloroacetic acid. So you can use both. You can use the formal name or the common name. They're both well understood by chemists. Butanoic acid, butyric. And butyric comes from butter. That's rancid butter fat will produce butyric acid. It smells really bad. In fact, I don't even carry it in my organic labs anymore because if it gets spilt, it's just too stinky. It's easier to work with acetic acids for those types of reactions because if it gets spilt, it just smells like vinegar. And people in general aren't completely repulsed by vinegar, so it just works out a lot better. Hexanoic acid is caproic acid, octanoic acid, caprylic acid, and decanoic acid, capric acid. Notice these are all coming from goat fats. Goats have much shorter chain fatty acids than a lot of animals. Like beef have a much longer chain fatty acid. That's 18 carbons, octadecanoic. And that's stearic acid. Or from the palm, palmitic acid, because it's from the palm. Hexadecanoic acid, 16 carbons. Solubility of carboxylic acids is determined by two groups. How big the nonpolar part is, and then you have the COOH group. So there's 1 to 4 carbons, so formic acid or methanoic, acetic or ethanoic, propanoic or butyric acid, it'll dissolve in water. 5 to 7 carbons is slightly water soluble. Some will dissolve into water, but you'll have 2 layers overall. And then 8 or more carbons, it does not dissolve in water, and the nonpolar group is too big. There's a lot of hydrogen bonding in carboxylic acids, which gives them very high boiling points. There's a lot of intermolecular bonding because you get twice as many hydrogen bonds as we saw in the alcohols. And we have acid-base chemistry. Here, water is absorbing a proton and becoming hydronium ion. Here, acetic acid, this proton is dissociating away, and you get acetate. Phenol, this proton can pop off a little bit, and you get phenoxide. Phenols, though, are much weaker acids than carboxylic acids, but still, weak enough where sodium hydroxide could easily remove that proton. So you do have a small percentage of protons popping off. And that is signified by these double arrows. You notice that bigger back arrow, they're signifying that most of the molecules don't dissociate. Only like 5% of acetic acids actually dissociate into ions. The other 95% stay intact, so most of them stay in a molecule. But 1 of every 20 or so, that H plus pops off like this. Carboxylic acids in biochemistry. Carboxylic acids are released into the blood by the liver in excessive amounts during ketoacidosis, which is a condition associated with uncontrolled diabetes. Normal amounts of carboxylic acids can be controlled by blood buffers and by the respiratory rates and kidney functions of the body. If too many carboxylic acids get into the blood, now we have acidosis and the blood's getting too acidic, and that is a pretty serious emergency. It needs hospitalization. L-DOPA, levodopa. Here we see this compound. It's a carboxylic acid used in the treatment of Parkinson's disease. It's a prodrug for dopamine. And in the body, the carboxylic acid group gets removed, leaving dopamine, which is an essential neurotransmitter for many functions in the brain. Major types of carboxylic acids. You have unsaturated, just straight single bonds. Aromatics, like benzoic acid. Dicarboxylic acids, like oxalic acid. Oxalate is what kidney stones are made out of, and so it's found in large amounts in rhubarb leaves, which is why you eat the stem of rhubarb, but you could never eat the leaf of rhubarb because they're poisonous, because they have oxalic acid. Here we have hydroxy acids. This is an alpha hydroxy acid. Again, it has the OH, the hydroxy group, in the alpha position, like lactic acid. And then there are amino acids. So we had saturated carboxylic acids with no double bonds. We had unsaturated carboxylic acids that do have a double bond. That's why it's unsaturated. And all these other types. Unsaturated carboxylic acids contain one or more double bonds, or can have a triple bond. The double bond is actually more effective because it puts a kink in the structure. So here we have stearic acid, completely saturated. This would be a saturated fat, and melting point is 70 degrees Celsius. Oleic acid is just as big, 18 carbons, but putting one double bond in there lowers the melting point down to 16 degrees Celsius. Plant oils typically have lots of unsaturated carboxylic acids in there, and because basically in general a fat is a triglyceride. You have one molecule of glycerol and then 3 fatty acids attached to it. Plants typically have a lot of unsaturation, and so plant fats are typically liquid. So olive oil, canola oil, grapeseed oil, avocado oil. Plants have oils. Animals typically have saturated fats with no double bonds, and so it's a solid fat, like bacon grease is a solid fat. Aromatic acids. Here we have an aromatic group like benzoic acid, orthotoluic acid, metatoluic acid, paratoluic acid. Dicarboxylic acids have 2 carboxyl groups. And in these you can get an internal dehydration to form anhydrides. By heating it, we can remove this water here and connect them, leaving us an anhydride. Anhydrides are extremely reactive because if a water molecule bumps into them, you get 2 carboxylic acids, and that's water's everywhere in nature. So it's extremely rare to find an anhydride anywhere outside of a chemistry lab. And when you do find an anhydride in nature, it's because it's so sterically hindered, nothing can attack it. The alpha hydroxy acids, you have a hydroxy group there. We got lactic acid, it's found in sour milk as well as mussels after exercise. Because if you're working out so hard, you can't get enough oxygen to your cells, you start fermenting, and pyruvate takes over as the final electron acceptor instead of oxygen, reducing it to lactic acid. Salicylic acid, which is what aspirin is made out of, because that's acetyl salicylic acid. This is an aromatic acid. And citric acid. Here we have our tricarboxylic acid, and you have a hydroxy group there at the alpha carbon. It's extremely common in citrus fruits, and it can make up 5 to 8% of lemon juice, which makes it very acidic. Here we have malic acid and tartaric acid, which is found in grapes and apples. Amino acids, this is what proteins are made out of. It's called an amino acid because you have both an amino group and you have a carboxylic acid group. And you have a hydrogen always, and then you have an R group. And what separates all the amino acids is the identity of what that R group is. And in general, you start with 20 amino acids, which are coded for by the human DNA in biology, but then you keep building up from there. And there's actually lots more than 20 amino acids, which are biologically significant. But the only difference is what the R group is. So glycine is the simplest amino acid. R is just H, and that makes it the smallest amino acid. All right, so here we have a dicarboxylic acid with 3 carbons. So you have to have your 3 carbons. And then it's a dicarboxylic acid, so you have 2 carboxylic acids, 1 on each end. And again, you can have more than 2. As we saw, citric acid has 3 carboxylic acid groups. You just have to have at least 3. All the carboxylic acid groups are primary carbons because they're only attached to one other carbon, because the carboxylic acid is 3 bonds in and of itself, so it can only be attached to one other carbon. And therefore, as long as you have as many primary carbons as you have, that's how many carboxylic acid groups you could theoretically have. Alpha amino acid with 3 carbon atoms. Again, we have our 3 carbons. And at the alpha carbon, we have an amino group. So this is 2-aminopropanoic acid, or alpha-aminopropanoic acid. How to prepare carboxylic acids? We can oxidize alcohols or aldehydes, or we can oxidize groups on aromatic rings, or we can hydrolyze nitriles. So here is the oxidation of an alcohol. It's got to be a primary alcohol to a carboxylic acid. It must be a primary alcohol. This will not work with a secondary alcohol because you're not going to break the carbon-carbon bond there. Here we oxidize benzyl alcohol to a carboxylic acid. Here we oxidize an aldehyde to a carboxylic acid. And that's a relatively easy reaction because aldehydes oxidize extremely easily. In fact, benzoic acid benzaldehyde oxidizes rapidly to benzoic acid just in the presence of oxygen. So it's very easy to oxidize an aldehyde to a carboxylic acid. And again, when you oxidize a substituent on an aryl group, you lose everything past your first carbon. So it's always the first carbon that becomes the carboxylic acid group, and it doesn't matter how long that chain is, it'll all be gone and ripped off in this reaction. And we can hydrolyze nitriles. Again, nitriles are R, C, triple bond N. Acetonitrile is a common solvent because it's very polar and there's no protic hydrogen, so it's very useful in organic chemistry. So what primary alcohol can be oxidized to form pentanoic acid? 1-pentanol. It's got to be a primary alcohol to become a benzoic or a pentanoic acid. So only primary alcohols can be oxidized into carboxylic acids. 1-chlorobutane reacts with potassium cyanide. Then the product is hydrolyzed to form a carboxylic acid. Well, first, Cn- from the cyanide will displace the chloride. And so now we gained 1 carbon. We went from 4 to 5 carbons. So we started with butane, but now we have 5 carbons. And when we oxidize that and hydrolyze it, so water attacks, we get pentanoic acid. So it's important to remember when you attack things with cyanide, you actually gain a carbon in the carbon chain, which is why it's so useful. It's one of the most convenient ways to add a single carbon to a growing carbon chain. So even though it's very toxic, chemists still use it because it's very difficult to otherwise add 1 carbon sometimes.